Since I started biochemistry from the 70s, they began to understand lipid signaling. I remember going to many lectures on it from visiting academics from all over the world. It's an exciting and important area, but I think with each 10-year period, there's new signalings being discovered, and refinement of that is happening. Of all my decades doing research, that was one of the nicest projects, most exciting projects, most flabbergasted projects I've ever done. Hi there, my name is Richie Porter and I'm a professor in biochemistry here at Trinity College Dublin, and I'm also currently the Dean of Students here.
Hi, I'm Saoirse O'Sullivan and I'm the Vice President of Translational Sciences at Artelo Biosciences.
As I said, I work in the area of metabolism, and one of the key signaling areas in metabolism, as you know, is lipid signaling, and it manifests itself in nature in many different ways. So a classic example might be a hormone hitting a receptor, G-coupled protein receptor, that then triggers an event that causes the release of lipids from membranes. Phosphate being released, again, it has a signal transduction, cell survival role within the cell. In the context of cannabinoids then, again, going back to triggering an event, phospholipase C might cleave fatty acids to release diacylglycerol, and that can be then covalently modified into an endocannabinoid, for instance. So most of the endocannabinoids that exist are born out of the lipids that come from membranes.
Yeah.
And the triggering of their release from a membrane is a natural process, usually triggered by some sort of stimulus, whether it be a foreign stimulus, a hormone, or some other triggering event, stress event.
Do you think that lipid signaling and the role of lipids in the body is as well understood as maybe the role of proteins?
I think, since I started biochemistry from the 70s, they began to understand lipid signaling. I remember going to many lectures on it from visiting academics from all over the world. So it's an exciting and important area, but I think with each 10-year period, there's new signalings being discovered.
Yeah.
And refinement of that is happening. But from the point of view of what Artelo are looking at, these fatty acid binding proteins, that was totally new to me. I knew nothing about that till about
Even
5
though
years.
you'd been learning for years about lipids.
It was a source of endogenous lipids that could trigger events in cells that I had never even thought about.
Yeah.
And the fact that Artelo were looking at drugs that could stimulate the release of those lipids from the fatty acid binding proteins to mitigate against cancer, to mitigate against type 2 diabetes, to counteract pain. I'd never even thought about that before. So that was very exciting to me.
Yeah.
And I had previously just thought of fatty acid binding proteins as a transporter of lipids, a sink for lipids. But the research that I did with Artelo opened my eyes to the specificity of them and how they could be, manipulated for the benefit of our health, I suppose you could say.
And do you think that lipid modification, which is what we talk about in Artelo, do you think that's quite a unique strategy in pharmaceutical companies? Are there many other medicines out there that are looking to modify the lipidome?
I'm not aware of any. No, there may be, but I'm not aware of that. No, not at all. Most drugs are focused on the exterior of the cell.
Yeah.
They're focused on, they're mopping up stuff with antibodies, triggering receptors with antibodies, small molecules that interact with the receptors. But no, I've never, in my experience, seen a focus on modified fatty acids before. No.
And part of the problem that we come up against is that it's a complex system to try and
Mhm.
explain as mechanism of
Yeah.
action for a drug, as opposed to a receptor agonist or
Correct.
antagonist. It's a
Correct.
very simple
Correct. Yeah.
story.
Yeah. Yeah.
Here's a small molecule,
Yeah.
it binds to this receptor or it blocks this
Yeah.
receptor,
Yeah.
therefore QED. But lipid modification is such a broader
Absolutely. Yeah.
kind of concept
It
of
is.
changing the biology
Yeah.
of our cell. And
Yeah.
we definitely find that something that's harder to describe to people.
Sure. But having said that, as I alluded to earlier, like endocannabinoids are modified lipids.
Yeah.
So nature does that, and we're just encouraging them to do it more, I suppose you could say, or
Yeah.
we can manipulate the equilibrium that's there. So lipid modification occurs within us to create crucial molecules, and we're trying to direct how that happens
Yeah.
via interaction with fatty acid binding proteins. And luckily enough, as you may or may not know, well, you do know, of course, but fatty acid binding proteins there's a variety of them. They're a well-conserved protein through evolution, but there seems to be some specific ones for specific tissues, and that also then makes the targeting our drugs tissue-specific. We could
Yeah.
have a heart and muscle specific drug, epidermal cell specific drug, or whatever, liver specific drug. So that also is a layer of what you could say refinement that gives us great potential for our drugs.
Yeah, indeed,
Not shallow.
indeed. So you've mentioned fatty acid binding proteins a couple of times now. Should we go back a step and really talk about what they are? Because they're a key tool in Artelo's discovery platform. So just at basic level, what is a fatty acid binding protein?
Well, again, born out of my ignorance before I started Artelo, I always associated fatty acid binding proteins with the outside of cells because they are in our circulation, they are outside the cells. But I subsequently learned there's equivalent fatty acid binding proteins within cells. They're small proteins, 14, 15 kilodaltons.
Yeah.
But they have a structure that allows them to bind. The structure is lipophilic, so they can bind lipids. So it's like I suppose you could say, like a docking station for certain lipids dependent on the fatty acid binding protein, of which there are roughly 10 in humans. But as I said, they're also tissue specific. So certain lipids are bound to certain fatty acid binding proteins. So they're sort of, what would you call it, banks of fatty acids and banks of lipids.
I think of them as little dodgem cars.
Yes.
And inside every little dodgem car, different lipids can get in and then get moved around. That's how I think about it in my head.
I would see it like that too. And, certain lipids can only bind to a certain fatty acid binding protein, not others.
Yeah. So some lipids can only get into some cars and not other cars.
Exactly.
Yeah. And where did the cars go? Like once those lipids are bound into the protein, what happens to them? Why are they binding in the first instance?
Well, I would argue it's a bit like bovine serum albumin in the circulation. It binds fatty acids, so it's transported around, and as it gets close to tissues where you deposit fatty acids, there's deposition there.
Yeah.
So at one level, that exact same thing happens inside your cells. So there are triggers that will naturally trigger the release of this storage facility of lipids to do whatever it needs to be done. So they might be released to trigger PPAR receptors in the nucleus, or they might be triggered to, hit other receptors. So they're like a bank of specialized lipids that can be tapped to release them. That's the way I view it.
Yeah.
And so the point is lipids can't hang out in the aqueous phase. They need to be bound to these structures. So that's their job. Their job is to be the sink. Yeah. A very specific sink for particular lipids. That's the job of the fatty acid binding proteins.
And we both know, and especially I know about the endocannabinoid system.
Yes, yeah.
What's the link there? So what is the link between endocannabinoids, which you've already mentioned are fatty acid derivatives, and these fatty acid binding proteins within the cell?
Well, again, it's because endocannabinoids are modified fatty acids, essentially. they will have a site on fatty acid binding proteins to bind. So therefore there could be a sink of endocannabinoids that can be released via natural processes or the drugs that Artelo develop. So my view is, again, there are clearly fatty acid binding proteins that have preference for endocannabinoids, and therefore it's an intracellular source of them.
Yes.
And they could be tapped in a natural way, or they could be tapped by drugs.
Yeah.
But the point being was, until this, I never knew there was such a storage for lipids. I didn't know there was even a preference for endocannabinoid storage. So that's wonderful to work in such virgin territory, really.
Yeah, and that is the great thing about working in lipids and in endocannabinoids is that, as you mentioned, every 10 years we're learning more and more, and then how we apply that science to new therapeutics.
Yes.
We're really working on the cutting edge of new science.
I would totally concur with that, absolutely,
So you've mentioned already that there isn't just one fatty acid binding protein. We actually have different isoforms, like different types of fatty acid binding proteins. And I think there's about 9 now, is that correct?
That's right, 9 or 10, and they are different. They're different in mass, but they're different in specificity. But also were identified in different tissues. So, for instance, fatty acid binding protein 5 has always been associated with epidermal cells, as an example. Fatty acid binding protein 3 with heart and muscle. But having said that, there is overlap. They do appear in other tissues. And interestingly, from the point of view of let's say, cancer therapy or pain targeting, in cancer, there's an increase in the abundance of certain fatty acid binding proteins in certain tissues, which makes them a particularly interesting target.
Yeah.
And as I said, I've worked with you on some of the Artelo drugs that inhibit fatty acid binding protein function. But maybe you could tell us the current thinking about the mechanism by which the Artelo drugs work, maybe in the analgesic space, for instance.
Yeah, of course. So we have quite a library of fatty acid binding protein inhibitors.
Right.
So we have ones that are selective for some of the different isoforms.
Right.
So particularly we've got FABP3, FABP5 and FABP7 selective inhibitors. And we also have some inhibitors that are dual inhibitors. So they might hit 5 and 7 or 3 and 7. And then we have some which are pan inhibitors. So they inhibit all of the isoforms. And you've mentioned that some are more important in some diseases than others. And that's why we have such a library of different molecules. But our lead compound, so the compound that we're working on and we've actually brought to clinic, is an FABP5 inhibitor.
Okay.
So it's a selective and potent FABP5 inhibitor that we developed with Stony Brook University because they were the first to really discover that fatty acid binding proteins were also transporters of endocannabinoids.
Sure.
So they started this space.
Sure.
And when they realized fatty acid binding proteins were endocannabinoid transporters, they thought, well, does this mean that if I inhibited that transport, that I could get some cannabinoid-like effects within a body? And probably one of the best known cannabinoid effects is analgesia.
Yes, very good.
So they basically showed through a whole series of experiments that by inhibiting that transporter, the little dodgem car, you actually prevented endocannabinoids from being degraded. So they're normally broken down by an enzyme inside the cell. And FABP brings the endocannabinoids to that enzymes to be broken down. So if you stop that, you increase the amount of endocannabinoids that are around.
So you're boosting the levels of endogenous cannabinoids.
Yes. It's actually,
again, what a wonderful way for a drug to work to boost the natural endocannabinoids.
Yes, enhancing what's already there.
Yeah.
And other people have tried to do that by blocking the enzyme itself.
So the enzyme that breaks down the endocannabinoid.
Yeah. So that enzyme is called fatty acid amide hydrolase, or FA.
FA, yeah.
And people have tried to just specifically target that.
Yeah.
Whereas we're targeting the bus that brings the endocannabinoids to FA.
Very good.
And what we with Stony Brook showed was that if you do that, FABP5 selective inhibitors are very good at relieving pain.
Very good.
Yeah,
Very good.
and that mechanism of action is through cannabinoid receptor activation. So it's
Fantastic.
through activation of the CB1 receptor,
Sure.
and other targets that we know cannabinoids or endocannabinoids work through, like TRPV1 channels. That's
Yes.
the chili pepper channel.
Yes.
That's
Yeah.
very important in pain sensation.
Okay.
So we've shown through all our preclinical experiments that FABP5 inhibition is a very good analgesic. And now we're moving that compound to the clinic, and it's called ART26.12.
ART26.12.
Yeah,
Okay.
so that's the name of our lead compound.
And what sort of patients would be taking that drug then? What patients need that drug?
Yeah, well, I mean, I think in the chronic pain space, you'll find that there's a real unmet need for new medications. There are meds out there for chronic pain,
Sure.
but they either don't always work very well,
Sure.
or they have quite intolerable side effects, particularly in the CNS. So
Sure.
sedation, memory
Yes.
loss, lots of
Yeah.
significant side effects with those analgesics. So we've been very fortunate that so far in our human trials, R2612 has shown a very, very good side effect profile. So we're seeing
That's
very,
great.
very few what's called adverse events. So that's just side effects associated with the drug in healthy volunteers. And we're not seeing any kind of signs of CNS disturbances. So no changes in memory or sedation or time perception, any of those things that you might monitor with a new drug, we're not seeing with R2612. So we're seeing a really nice side effect profile so far with the drug. And there are many indications for which that could be used. But particularly we're interested in the pain that's associated with chemotherapy treatment. So people who undergo chemotherapy often develop what's called peripheral neuropathy, or might be associated with pins and needles, a loss of sensation, or pain, stinging sensations. And so we're particularly interested in the ability of our drug to either prevent or treat chemotherapy-induced peripheral neuropathy, which is a real significant unmet need. And our strategy, our company strategy, is very much around cancer and cancer supportive care.